All right. Welcome
Ben,
back.
welcome back. Welcome back.
Thank you.
How's the jet lag treating you?
I'm actually doing okay. I tried to do my very best to stay up late while I was in Europe so it wouldn't completely destroy me
It's
when I
probably
got
very
back.
easy
Yeah,
staying
yeah.
up late like that. Whenever I go to the West Coast, I'm always up at like 4 a.m. And it's just like, well, I'm up now. This is how things gotta be.
Excellent. Yes, so I'm doing well. Perfect. All right. Well, everyone, welcome to another episode of Bio to Bedside, where we took a little bit of a hiatus during the early part of October, but we're now back with a vengeance. And we are quite excited for the conversation today. We're going to be looking at an allogeneic approach for cell therapy. Obviously, that's a fairly hot topic right now. A lot of different moving inside of the cell therapy space, both with autologous and allogeneic applications. And so very much looking forward to the conversation later today. Just as we get started here, again, we want this to be a conversation as much as possible. And so if you have any questions for our guests once we bring them in, by all means, feel free to put a comment down below and we'll try to get to that as quickly as possible. So just as we're getting started here for the first couple minutes, we always love to hear where people are coming in from across the world. So if you can just leave us just put a comment down below, let us know where you're coming in from. It's always cool to see how we get people coming in from all across the world. And yeah,
all your Polish and German friends that Ben made last week.
Oh,
I'm not jealous at all, if you couldn't tell.
We'll get you on Polish trip 2.0, Jeff.
Yeah. Sounds good.
But yeah, it is pretty cool to see for a company like, well, not a company, a country like that, just how far they have come. You know, obviously, 35 years ago they were communists, and the Soviet Union fell, and there's been some pretty fantastic growth since then.
Development since. Nice.
Perfect. All right, shall we bring in our guest, Jeff? Do you want to introduce where these folks are from and what they do?
I can do my best. I think, when I first heard of Genicity, I knew them as an Irish company, but I'm not super sure if that's still the case. We're going to find out. But yeah, they're doing an allogeneic T-cell therapy, but it's very, very, very different from the majority of allogeneic T-cell therapies that are on the market or in development right now. I'll let them do a lot of the talking, but basically what I thought the company was doing at first was making the first allogeneic T-cell therapy that was compatible with T-cell engagers. Because most allogeneic T-cell therapies have to delete the TCR to make them safe to infuse. Otherwise, if you had a TCR, you'd get graft versus host disease and it would be like deadly, basically. So by making the T cells safe for infusion, you actually make them incapable of binding to T-cell engagers because T-cell engagers require CD3. And CD3 is a part of the TCR. So you delete the TCR, you get rid of CD3, and the T-cell engagers can't bind to the cells anymore. But I talked to him a few months ago, and it's actually significantly deeper than that. It does do that as well, but the biology is super, super interesting. So I'm very excited to have them on the show today.
Well, let's bring them in. Perfect. All right. First off, we have Rainer and then Munir. Gentlemen, can you please introduce yourselves? And oops, just a second here. And what you do, we'll put you both on top there. So you got the hot seats. Sorry, and I talked about this right before the call. So my English pronunciation immediately butchered. It's Reiner's name, not Rainer. But the phonics in me just immediately destroyed it. So apologies for that. Excellent. So yes, we'll start with you, Rainer. Can you introduce yourself and what you do?
Yeah. Very happy to be here. And thank you for the invitation to talk about Genicity today. I'm in the industry for quite a number of years. I'm with Genicity since a year and a half. Joined the company in February last year. And I got to know the founders, Munir and his colleague, Muhammad, about 2 years ago through a mutual acquaintance who met them at an accelerator workshop and said, what these guys are doing is really exciting. And with your experience in the industry, they might benefit from that. So I got to know them and got excited about it and decided that I'll join the company and bring the platform all the way to clinical trials as we are setting out to do.
Fantastic. All right. And Munir, please. Can you hear us?
Sorry, Ben, if you are talking to me, I cannot hear you. I can hear everyone else.
Oh,
Oh.
my goodness.
All right.
Yes.
Well,
That's
Jeff,
not going to be good. Well, if you can hear me, introduce yourself for the audience.
So it's a pleasure to meet you. Jeff and Ben, thank you for having us in your platform. It's a pleasure and an honor to introduce our technology and our unique thinking about allogenic cell therapies and how we are addressing the limitations, especially in solid tumors. My name is Munir. I am one of the co-founders of Genicity. In my background, I hold an MD degree. I had 2 years in partial residency in general surgery and oncological surgery. And since the inception of the idea and day one of the technology, my partner shared it with us. I was amazed by the potential of what we can do, and we have been in that journey since then. And as Rainer has mentioned, as a talent that have no experience, we had someone to lead the ship, and we are honored, and we were very lucky to have Rainer as our CEO, with more than 40 years in the field of the biotech, and even having approved drugs and marketed drugs under his name. So we're very happy, and we're happy to introduce ourselves to you today and tell you about our unique idea.
Awesome. Well, maybe Jeff, I'll get you to leave the conversation a little bit with regards to talking to Munir here. We just got a bit of a technical issue with him hearing me. But yes, I guess to get started here, Rainer, can you tell me a little bit just about the problem here, right? What's the major problem that's getting tackled here with Genicity?
Sure. Of course, we all know that the onset of immunotherapy in cancer has been a major game changer. And it all started with checkpoint inhibition, with CTLA-4 and PD-1 inhibition, and got expanded after that with CAR T cells, and most recently with the bispecific T cell engagers. All of those are dependent on the patient's immune system to get activated against the tumor. And it's most specifically, the T cells are the major players here. And while these approaches have been a game changer, there are major issues that are still unresolved. And those issues are mostly related to insufficient efficacy and the number of non-responders, especially in solid tumors. And this is the area where we believe our ENABLR platform will bring change and will bring in a new paradigm, and allow especially the use of bispecific T cell engagers in the field of solid tumor.
So can you speak on some of the statistics around that? There is a lot of data to suggest that the failure of T cell engagers is actually not due to antigen escape in some cases, and then it's suggested that it's a large proportion of failure is actually due to exhausted T cells or insufficient T cell activity. What are some of the actual statistics to back that up? I've seen them around, but can you speak to that?
Sure. So all of the approved T cell engagers, with the exception of tarlatamab, which is approved for small cell lung cancer, and with tebentafusp, which is for ocular melanoma, are in the field of hematological tumors. There, the overall response rate is in the range of 50 to 60%, with a deep response in around 25 to 30% of patients. But even those patients which have an objective response, in many cases relapse. And this relapse is partially due to antigen escape, but the majority of these relapses, and also of the primary non-responders, is due to the T cells of the patient being in what's called an exhausted state, which means that due to the sustained exposure to antigen, in this case tumor antigen, the T cells go into a state of down a trajectory of epigenetic reprogramming, which leaves them in a position to be, so they cannot be activated anymore, and they've lost their cytotoxic, basically lost their cytotoxicity.
Right.
And in solid tumors, the experience with tarlatamab is that the overall response rate is around 30 to 40%. That's also true for the newer ones like obrixtamab from Boehringer Ingelheim, which is in clinical development, which in early trials shows a similar overall response rate. So there is 60% of patients who are not responding and who are in dire need of support to their immune system, which we hope to bring with our ENABLR platform.
Now, do you have any primary indications that you're focusing on first, or is it just kind of like, you know, T-cell engager combinations across the board?
No, we go basically in our data in the lab, we have shown that basically all T-cell engagers that we could get our hands on are activating our enabler cells.
Nice.
But we want to go along with T-cell engagers that are already on the market, and this is most specifically tarlatamab or imdeltra from Amgen,
Sure.
where we can add our cells to an ongoing course of treatment with imdeltra once the treating physicians notes that the patient is not responding.
Now, how long does that normally take? And is that a complex process?
Well, no, the patient gets a prescription of the T-cell engager, and after the updosing part and the first treatment cycle with full dose, there is an assessment of efficacy.
Sure.
And when this assessment shows that the patient has either progressive disease or stable disease, then this is an indication for adding the enabler cells.
Yeah. And is the Amgen, I'm trying to look it up here, is the Amgen engager for solid tumors?
Yeah, it's for small cell lung cancer.
Yeah.
It's the first, after the very special case of tebentafusp, which is for ocular melanoma, small indication. This is the major approval for an important solid tumor indication.
Right. Now,
can I jump in and ask a question here? This is quite interesting to me. Just a question for you, Reiner, would be, how about, so why is it that people haven't done this before? It kind of seems, it makes a lot of sense, right? If I understand correctly, you've already got an existing T-cell engager. Why not also give it a bunch of T-cells at the same time? So you have the two pieces of the equation instead of just one. Why is it that this hasn't happened before?
Of course, this is a logical step to consider. However, if you add a healthy T cell from a donor, you have to cope with the problem that these are allogeneic cells. So they have a T cell receptor which recognizes antigens in the patient, which leads to alloreaction, which leads to graft versus host disease. And this can be, as you mentioned in the introduction, can actually be fatal. Then one could think of, and this is what companies in the allogeneic cell therapy space have done, remove the T cell receptor completely. But then with bispecific T cell engagers, obviously the cells cannot be activated anymore. So we needed to design a T cell receptor while being not being able to recognize antigen, still have the full capacity to be activated through the CD3 complex. And this is not so easy as it seems, because if you take away the recognition sites of the T cell receptor and shorten the alpha and beta chain, the T cell receptor tends to not being transported from the ER to the cell surface.
Yep.
So we had to do some additional editing to and mutations to the T cell receptor that, without the recognition site, it's still expressed on the cell surface. So this is part of our innovation process.
Yeah, it's very finicky in that sense. It's very sensitive to any kind of manipulation, and the whole like T3 complex with the alpha, beta, and the CD3 subunits, it needs to be co-expressed. And in fact, it's funny. I had somebody message me a few days ago about, hey, can we express? They're talking about targeting lipid nanoparticles or AAVs to specific cell types, and they asked me, why can't you just implant like a TCR onto the surface of a lipid nanoparticle or an AAV? And I was like, well, for many reasons, but it got me thinking again. I was like, it would need to be alpha, beta, and all of the CD3s
and
all there, because it needs to have the whole thing. Yeah, so it's not just like expressing the alpha and beta to get the recognition. You need the whole thing. Otherwise, you're not gonna. It needs to be like there as a unit. And yeah, like you said, if you just chop off those recognition domains, it never gets to the surface. So you might as well just delete the whole thing, right?
Yeah.
So yeah, and in fact, a lot of the edits for allogeneic T-cell therapies is just a track 1, which is the alpha deletion, and the whole thing's gone.
Or you could also delete the beta. One of the two will delete the whole thing. Yes.
Yeah. So but I have a question. And this might be jumping ahead a little bit, but I want to. Ben, you asked, why hasn't anybody done this before, where you have a bolus of T cells being infused that are able to recognize antigen via an antibody recognition capacity. That's what CAR T cells are, right? CAR T cells are a bolus of T cells that recognize an antigen with an antibody fragment. So the question becomes, what is the difference between a T-cell engager plus allogeneic T-cell therapy versus just a CAR T-cell therapy? What is the added benefit of having a T-cell engager like mediated activation versus just a CAR?
Yeah. So Munir, you want to take that one?
I hope you can hear me well. When you look at the CAR Ts, one of the things that once we started our company and the idea of having allogenic cell therapy is, how can we have off-the-shelf cells that can be given to any patient without the need to have manufacturing again, and have all of the manufacturing streamline dedicated to one person or even few patients. The idea was behind it, if you will have cells, even an allogenic cells that express CAR T, it will be specifically for specific indication. And once you have a loss of antigen in the patient, you need to have additional CAR Ts for a different antigen. Our idea was, why not to split the cells from the recognition? Why not have cells that can be allogenic, ready all the time, that can be given to any patient, and only have a switch on, switch off mechanism, which was already there. When we started our idea, we wanted to think about our own kind of mechanism of switch on, switch off. And we succeeded, as Jeff said. It's very hard to express again the CD3. And we succeeded to do it. And once we have seen the CD3, we say that there is already approved T-cell engager. Why not to use them? And we have seen that the exhaustion problem of the T-cell engager. So we have a solution that fits for everyone. When you work with the CAR T, usually the signaling mechanism and the activation mechanism is different than the TCR. It's not the normal T-cell activation, which usually leads... Please feel free, Jeff.
Well, it's synthetic. It's not... We don't express CARs normally, right? So T cells, it's like strapping like a laser beam to my shoulder. Like, it will do it, but it's not natural. It's going to hurt me, right?
And we have seen it even in our dishes in the lab, that the CAR Ts behave differently in terms of metabolism, in terms of signaling, in terms of activation, in terms also exhaustion. So you have T cells that are overactivated. We don't want it. Both the founders, we come from the world of medicine. We are practicing medicine. And usually as a doctor, we like things simple. So we like to do as less as manipulation for the cells and keep them normal as possible, because, well, normal T cells can attack tumor. They can do it without any problems. But you need to have a trigger, the recognition. So for us, the combination was from our cells, which are totally blind, that can be combined with any T-cell engager, was the ultimate solution. We can have our cells, and we can add any T-cell engager that we want for the specific indication. Why is that important? First, you will not see the same exhaustion that you will see in the CAR T. It's the normal mechanism that you are having the killing for the tumors. Also, it will be seen in the side effects in terms of the cytokine release syndrome, because it's the normal way of attacking things. Well, when we get sick, any one of us, simple things, you will not have a CRS as the CAR Ts.
Sure.
The last thing is related to the costs. If you have a streamline that you have a process for the same cells and you will have it off the shelf, it will reduce the costs massively. And then you will shorten the time from, as we say, from needle to needle, from the donor to the patient. And you will have the luxury of choosing the best donors for the patients.
Right.
Yeah, and there's an additional issue which we are tackling with the same sweep, but more or less is the issue that all allogenic CAR T companies are facing, and that is the poor persistence of the allogenic CAR T cells. They typically are gone when you look, they are gone within 4 weeks. And even when companies like Caribou and CRISPR and Sana try to prevent the disappearance of the CAR T cells by making them kind of immune cloaked, that doesn't help. And because they still disappear with when you knock out HLA-1, HLA-2, overexpress CD47. So the issue, that's why this is really the case, and more evidence is coming up in the literature. It's actually the lack of the T-cell receptor that makes the allogenic CAR T cells non-persistent. And there's a lot of evidence showing that there is a basic signaling from the TCR, even when it's not engaged with antigen, that makes the T cells healthy and leads to their persistence.
That's super, super interesting, and I say controversial. That may be, there is an entire industry of technology. I'm thinking of Shinobi, Sana, Century, all kinds of stealthing technology. Mm-hmm. And granted, the major focus of a lot of those companies is allogeneic T-cell therapies, but Sana does islet cell for type 1 diabetes. Yeah. There's other kinds of hypoimmune technologies for things like organ transplantation even. So it isn't all about T-cell engagers or T-cell therapies. But what do you say about, something like an NK? Or I'll say this. There still is use for the stealthing technologies. It isn't just the T-cell TCR deletion that makes them non-persistent. You kind of have both of those, is what you're thinking, because they still will be targeted and rejected by the host's immune system if you don't stealth.
There is not a lot of evidence that de novo MHC antibodies are actually induced when you put in allogenic T cells. So the stealthing works beautifully, as you mentioned, for islet cells, for beta cells. They persist for more than 12 months and continue to produce insulin. But the SC219 product from Sana, which is the same technology applied to a CAR T cell, actually doesn't have that same effect. They're gone very quickly.
Yeah. So
the stealthing helps. Stealthing helps for certain cell types, but it doesn't help apparently for immune cells.
Could it? I mean, this is just me spitballing now. Could it be due to the differences in the trafficking of those kinds of cells? If you were to infuse T cells in intravenously, they're going to go to the lymphatic system and be targeted relatively quickly. So you need to have, they're more vulnerable to being attacked, maybe, and that's why you see the discrepancies. Maybe, I don't know.
Munir, you want to comment on that?
So when you look at the problem of the persistence and the durability of the T cells, one of the major things that we have seen day one in our lab is the lack of the TCR. So for us, it was very important to have the TCR, and successfully we achieved to have a complete TCR but without any recognition. The other arm that we are also thinking about, which also key opinion leaders and people like you, Jeff, asks, well, what will happen if the recipient attack your cells? Here comes the HLA matching. And the most recent data for allogenic companies, they do a partial HLA matching. As an example, if we give you, like if you look at New York with the variety of the ethnicities and everything, we can have 10 to 20 donors from New York to cover all of New York. And when you have a partial HLA matching, then you diminish and lower the percentage of having what we say the host versus the graft, not
Sure.
the other way around. So that's our way of thinking how we will tackle this issue. In addition, in our clinical protocol, we will have in our cohorts, 2 cohorts. The first is having a lymphodepletion, which is a standard of care of any cell therapies right now. And our thinking is the other way around. We want to try ourselves even without lymphodepletion as a second cohort to even see that our cells will persist and survive. But the most important thing that we all think about, do we want cell therapies and engineered cells to survive long in the patient's body?
Exactly. Yeah.
That's what we don't want.
Right.
So we wanted all the time, we thought, as a physician, we thought about the patient and then go to the science. So we need something very delicate that will survive long, but will not survive forever. So yes, we want the cells of the host to attack our cells, but not after 4 weeks, after a few months, after
Sure.
6, 8, 12 months, not before. And this is a delicate play between having the cells as long as possible to have the maximum effect, and not having a genetic kind of aberration that will affect the future health of the patient.
Interesting.
Jeff,
Okay.
why we are doing it? We want to help patients. We don't want to cause them any harm. Do no harm. That's the first ethical thing for us.
Sure.
So that's the idea.
Now, I want to push back a little bit just to see, like, because I'm a little bit naive in this topic. What is the potential risk of a persistent, base level of CAR T surviving for just kind of like doing nothing in circulation for 15 years? Who cares?
Rainer, I will mention a few things and then we'll let Rainer complete. But look at the CAR T CD19. Well, you will have a persistent depletion of...
Never mind. Yeah, never mind. You're right. I'm stupid. Okay.
Is that the genetic aberration that will have some kind of genotoxicity into transforming these cells into malignancies themselves, which is one of the new concerns that start to show and start to pop
up. So the CAR Ts have a black box warning for the rare cases of new malignancies from clonal expansion of the added cells. And we want to avoid that.
Very good. Okay, Jeff, I might ask you to take the lead on questions just because Munir.
Oh, sure. Well, I'm going to pop up this one real quick because we actually came to this. So Chinrong asks, I have one basic question. Could TCR edits impair long-term T cell survival? Now, that's kind of your whole shtick, right? That's the whole thing. Yeah.
Exactly. Yeah.
So you actually want that.
Yeah, like you... The reason that Genicity exists is because they believe, and there's plenty of evidence to show, that TCR deletion impairs T cell persistence, which is why allogeneic T cells, at least partially, are not persistent, even with stealthing technology.
There's actually a group in China that has gone deep into this issue, and they found that there is the TCR deleted T cells have a very different expression profile of quite a number of proteins, up to several thousand proteins compared to TCR replete T cells. And there are a number of factors they have identified which are actually have been linked in the past with T cell survival and T cell fitness. So this makes a whole lot of sense also from single cell sequencing, which has been performed in this paper a couple of years ago.
Now, do you think it's like unknown biology? Do you have an idea as to what is actually going on with the presence of the TCR in the absence of antigen? The TCR is like doing something, right? Do we have any idea? I mean, not that it's like super important, I guess, but is there any idea as to what may be happening there?
Yeah, as I mentioned, this paper from the Chinese group in Cell a couple of years ago went into this issue, and they found that there are a number of factors that are overexpressed or repressed if you remove the T cell receptor. Some of them, and they're most two very important ones that belong to the transcription factors that drive the epigenetic programming of expression of factors in T cells.
Yeah, that's cool. I mean, see, the molecular biologist in me is like, well, what's going on down there? But at the end of the day, it's kind of like, well, we don't really need to know, Jeff. Just be quiet. We know that it helps to have it around.
We're actually very interested in that as well, to understand why this is the case. And there is some evidence, but it's not 100% clear what's going on in a T cell without antigen engagement, which makes the T cell survive long term.
Yeah.
One of the publication was at Lee et al. 2022 says that when you remove the TCR, there is a downregulation of two important genes, which are the NRR4E3 and the EGR3, which are very important for kind of survival of T cells. So this is one of the things that have been shown. And to be honest, the TCR has been a black box for others because everyone wanted to remove the TCR and no one thought about, well, how can it affect the T cells? Because they were afraid of the GVHD, and that's it. They didn't think it alone. And you can see till nowadays, that everyone is rushing to do other things, as Rainer has said, downregulate other things, express additional genes, CD47, CD52, HLA1, HLA2, beta-2 microglobulin to knock out. No one wanted to keep the TCR. I can't tell you why, because it's not easy to express the CD3. It wasn't easy for us, but we cracked it, and we did it with a lot of efforts. It's a delicate, complex, the CD3. Any minimal change, which we tried several genetic aberrations, several genetic modifications. The tiny thing, even the smallest amino acid, will prevent you from expressing the CD3. So,
yeah. And, to your point about one of the other, so that the retaining the TCR has those two massive benefits. The first one we've been talking about is the persistence of the T cells. There's this weird black box. The TCR is doing stuff there to keep them around. Not very sure what it is, but it's doing it, so we need it there. But the second benefit is obviously the ability to engage T cell receptors, and now we can give them eyeballs to go and look for stuff. And then you mentioned, a lot of people have just been deleting the TCR because to keep it around is really difficult. But there's companies and there's an entire field of immunotherapy that tried to kind of split the baby, and it's called universal CARs. And what they do is they have a universal Captain Hook arm CAR that can bind to proprietary or unique peptides that are engagers, but they're not T cell engagers. Some companies have tried to use antibodies. It's very difficult. So a lot of the times they kind of resort to these bespoke, antigen specific, unique peptides, which is very complicated to make. You need to make these binders, right? And at the end of the day, it's still just a CAR, and it's worse than a CAR because the binding of the engager to the receptor is not covalent, right? Where with you guys, I guess it's also affinity interaction, but you're not using a CAR. It's TCR signaling, which is very different.
Exactly. So, yeah. And the additional benefit, and you got that very well. And if you have a proprietary adapter molecule, that's a new biologic. So that requires a full development with toxicity and everything. Whereas in our case, we can use T-cell engagers which have already undergone the toxicity testing, and they're found to be safe. And in addition, and that's also very important, and it's an important detail, the T-cell engagers have a high affinity for the antigen, and they have an intermediate activity, intermediate affinity to CD3. So our cells, when they get into the patient and are in the patient, they don't see the TCR, the bispecific T-cell engager necessarily by itself, but they recognize the T-cell engager when it's already bound to the cancer cell and forms the immunological synapse and kills the cancer cell, and then it moves to the next labeled cancer cell, to speak.
Okay, so you can't. So you couldn't like pre-incubate.
We could, but it's not necessary. It's not necessary.
It wouldn't add any benefit.
It wouldn't have any benefit. No.
Yeah.
There are 2 points that we should mention. The first is you come down to the same problem of exhaustion of the T cells because you are activating them through an synthetic methodology. The other thing is the sensitivity of the TCR compared to the CAR. How many CARs you need to be engaged with the molecule in order to activate the T cells compared to TCR. And the TCR you need much less. Yeah. Because it's more sensitive.
Orders of magnitude.
Which allows you to capture antigen low cells. And it reduces the potential for antigen escape.
Exactly.
Yep.
One of our recent studies that we have done on hepatocellular carcinoma, we had mimicked a model of late-stage disease for patients with peritoneal metastasis, and we have used a cell line that have almost 900 to 700 molecules expressed of the antigen on the cell.
Yeah.
Which is very, very low.
Yeah.
When we combined our cells with the T-cell engager specific to this antigen, we had a huge reduction in the tumor mass and successful results. And we compared ourselves to normal T cells, healthy donor normal T cells, not a kind of a patient. So sensitivity, it's very important. Exhaustion, and we had a methodology that can combine ourselves, as Rainer mentioned, to already existing therapies that are safe in clinic. Every patient know their side effects, know how to work with them, and key opinion leaders, they like things that don't complicate things. They like to work with things that they are already familiar. Please make it better, and that's what we are trying to do.
So to that point, this question kind of asks a little bit. It says, what class of drug would this be? An antibody therapy, a cell therapy? The question that I have related to this is, does the FDA not consider it to be a new modality when you're combining a T-cell engager with an allogeneic T-cell therapy? Do they use the T-cell engager PK, PD, tox data, or is it a completely new type of thing?
Well, we haven't discussed that with the regulators yet. But from discussion with key opinion leaders, we believe, and we have data showing that that the dose response curves with our cells and with wild type unedited cells in combination with the T-cell engager is not different. So the EC50 of the T-cell engager is the same when you combine it with unedited cells and with our cells. So we don't expect something in exaggeration of toxicity or something like that.
Yeah.
However, we believe that, of course, for the first combination that we will try in the clinic, we will need an IND for this. But we also are convinced that once we have shown that it's safe in that combination, this has the potential to receive what's called by the FDA a platform approval.
Yeah.
So, and this has been platform approvals have been given so far to 2 LNP combinations for vaccines. But we believe that our enabler cells have all the ingredients actually for our platform approval. So once we have demonstrated safety and efficacy in one combination, we will be allowed to combine them with any T-cell engagers that's approved.
I would love to add to it, Jeff, that we had a discussion not with the FDA, we had a discussion, a pre-CTA a meeting with the European Medical Agency. And from their point of view, the therapy is an add-on therapy. So it's a kind of a combo.
Yeah.
Their suggestion was for us, in order to try a new therapy, they wanted us to combine ourselves with already approved T-cell engager.
Yeah.
As a company, we have the talent to develop our own T-cell engagers, but they clearly say to us, as initial phase 1 clinical trial, you need to see that your cells, in addition to the existing approved, well-known T-cell engager, which we know the side effects, does the addition of your cells affect it?
Sure.
And we had an advisor that we talked to. He was working with the big pharma and as a head of clinical trials, both in the cell therapy field and in the T-cell engager field. And he, from his point of view, he wanted to think about it as a booster, not as a therapy.
Sure.
Like it's an add-on. So as our idea is to have it as a booster, as an add-on, and as a platform, then we need to do, every time that we try a new T-cell engager, we don't need to do a full clinical trial. We need to do several patients, few patients, just to show the modality works and there is no change in the side effect.
Yeah, that makes sense.
Very good.
So one question here. This is going to the donor source. So CAR T persistence. What do you guys think about a concept of using cord blood derived T cells plus simplified donor host matching? Cord blood is much less immunogenic. Your
Shout
thoughts?
out, just one moment. I met Thomas in Krakow in Poland, so shout out to him for coming.
He's one of your new Polish friends. I'm jealous. So yes, cord blood derived, much less immunogenic, probably more available. Cord blood is everywhere, and normally it's considered medical waste. So what do you guys think of that?
Rainer, can I answer it?
Yeah, please go ahead.
That was the first question for ourselves. Like people will ask us, why not to use the concept of having partially matched normal T cells from healthy donors? That's why we added 3 elevation layers for our cells. Our cells are not only a normal T cells. They are not T cells that will be combined with T cell engagers. Our cells have their unique fusion protein and unique additional knockout of what we call our ENABLR gene. We make our cells to persist and to work in the most exhaustion and the harshest tumor microenvironment in order for our cells to work fully and perfectly. And I will let Rainer to explain it and add to that point. So we are not only a T cells, we are an addition to it. We are a unique cells that can work in solid tumors that have a unique phenotype of stem cell-like cells that can work in the hardest conditions and preserve stemness and eliminate as possible the exhaustion for the T cells. And Rainer will continue on it.
Yeah, so when we set out the design of the final enabler cells, we wanted to learn from what is actually really working, albeit in a minority of patients in solid tumors, and that's checkpoint inhibition. It's about 20% of patients respond well to checkpoint inhibition, or to the point that they get into long-term remission. But there's about 80% of patients that do not respond. And what differentiates these patients is a very unique phenotype of T cells, which is present in the responders, called the precursors of exhaustion. They are characterized by intermediate PD-1 expression and expression of a transition factor called TCF1. They are self-replicating, they're stem-like, but they are repressed from cytotoxic activity by their PD-1 expression and the engagement with PD-1 ligand. Now, when the PD-1 the anti-PD-1 antibody comes in, they're de-repressed, the brakes are loosened, and they can proliferate and become engaged with the tumor and kill. However, they also need they need co-stimulation through the CD28, which is they receive from dendritic cells, and patients who respond have dendritic cells in their tumor, and that's another unique feature of responders. We wanted, eventually we wanted to make our cells like activated TPEX cells, and that's why we express a fusion protein, which is made of 2 different cytokines with a flexible linker, which induces the stemness and increases the expression of cytolytic enzymes. And we knock out an intracellular checkpoint protein, which makes the cells resistant to PD-1 engagement, and also makes them independent from CD28 derived co-stimulation.
Yeah, because you're not going to get it. That's good. Yeah. So I was going to ask, with T-cell engagers, you're not going to get co-stimulation anyway. So normally you normally engage stem-like T-cells that don't need co-stimulation as much. But what you're saying is that your cells aren't going to need co-stimulation. So regardless of
need co-stimulation because of this knockout. Yes.
Regardless of their CD28 co-stimulation ability with the T-cell engager, which would be basically negligent, it doesn't matter. That's really good. I've always wondered that, and I always feel kind of dumb because the answer is probably out there. When you give a patient a T-cell engager, really the only cells that can engage that T-cell engager are memory cells, right? Because the naive cells won't respond to just CD3 engagement. It's anergic, right?
It's actually not memory cells, because in a tumor patient, there's not much. You get memory cells when you have an acute infection with a virus. Then the naive T cells get engaged, proliferate, and when the virus is eliminated, they convert to memory cells. In the constant in the prolonged stimulation with tumor antibodies, and also in chronic virus infections, you don't have much memory cells. The T cells are going down a different trajectory, and which is the exhaustion pathway.
But the patients, the patients themselves would have memory cells, and that's memory cells from prior infections and things like that, and that's normally the cells that would engage the T cell engagers. But the odds of those cells being in the tumor, or you're already limiting yourself to a pretty small proportion of the circulating lymphocytes already. So to have your cells, I was thinking, okay, you're gonna infuse, I don't know, let's say 1.5 million cells. Of those cells, only 15% are memory-like and can actually engage these things. But if you're saying, nope, we accounted for that, almost 100%, or a very high proportion of these cells are going to be able to engage with just the CD3.
Exactly. Yeah. So they have when they come out of our manufacturing process, they are to a very large extent CD8 only.
Yeah.
They are to a large extent stem-like cells, which resemble these progenitors of exhaustion, which are the effector cells in checkpoint inhibition.
Jeff, a unique thing about our cells is we have the naive stem cell-like cells. Usually and normally in the patient are other T cells. If you engage them with T-cell engager, they will not work.
Yeah.
But in our case, in our cells, because of the extra innovation layers and the fusion protein that we express and the additional knockout of our gene, our cells have a B-phenotypic model, which means that our cells represent and their all naivety and stemness. And they can become effector and start to kill the cells, the tumor cells, once they are engaged with the T-cell engager. That's why our cells don't need any other co-stimulation to process our cells. They shift directly to highly fully effector cells that target the cells and kill it. So you think about our platform as a pool of self-renewable cells that all the time have stem cell-like, naive-like cells that all the time provide you a new highly effector cells. And just one comment regarding the question that we had before about why not use a partially matched HLA T cells. There has been also in the recent years kind of choosing specific TCRs to have them from the patient even themselves or others patients or of healthy donors to recruit and expand the special T cells that are found in the tumor microenvironment and next to the tumor. But unfortunately, no success in it till now. We haven't seen any approved therapy of these. And the only thing that we have seen is the VSTs that are used not in the oncology, but in other indications. So that's it.
So I'm very interested in your thoughts on this. Any thoughts on this technology and how it can be used in Treg therapy? And you mentioned that your cells that you get are primarily CD8s. I'm going to guess that that's intentional. And the CD4 Tregs are probably, we're not worried about those.
No. Yeah. So,
right.
Yeah. So the process that our manufacturing process drives the composition to a large extent towards CD8 cells. So as Tregs are CD4, this really doesn't have an impact.
Because the question that I would have is maybe not so much on Treg therapy, but what is the potential of getting Tregs in your bolus of T cells, and then you're engaging the CD3 of Tregs, and you're actually suppressing the immune system. But if you have majority of CD8s, then not a problem.
That's not the case.
Yeah.
No.
And then the one last here. What about off-target toxicities? This would be very similar, I would assume, to just the general risk of T cell engagers or CAR T cells. There's not much difference.
Yeah, there's no difference.
No, there is Jeff, when we talk about the CAR T, there is a differentiation of the severity of the CRS and the tumor toxicity. Yeah. So we need to be very careful when we compare it to CAR T. No, the combination of our cells with the T cell engagers, which has been shown in animal models in vitro, we don't see, in comparison to normal T cells, higher cytokine release or interferon gamma. So our cells, in terms of CRS, we believe that they will be compared to the T cell engager itself. We may see a little bit higher, but we will not go up to the CAR T.
Yeah, sure. And also, the main driver of CRS are known to be the CD4 cells, not the CD8 cells. So the large increase in interleukin-6, TNF alpha, and IL-1 comes from CD4 cells and not from CD8 cells. Right. So the predominance of CD8 cells in our product limits the liability for CRS and ICANS.
And one comment about the Tregs, Jeff, is what about the Tregs that are in the patient immune system or within the tumor microenvironment? Can they affect our cells? The answer is no. Because the modification that we made for our cells, it kind of disrupts all of the exhaustion signaling. And the gene itself also cannot be affected when you knock it out. Our T cells cannot be affected by Tregs, or they cannot be suppressed by Tregs.
That sounds very interesting. I won't press too much, but that's, I mean, it seems, yes, when I first heard of the technology, I was kind of like, okay, yeah, there are some other companies who are doing similar things, but there seems to be something a little bit deeper here, where it's less about primarily being interested in making a cell that can engage T cell engagers, and it's more like we are preserving T cell activity. And we are trying to, we're working with the biology of T cells. Yeah, that's good.
So here's a question, Jeff, that you might have to repeat so Munir can hear it.
One thing is very worth mentioning.
Hang on, Munir, sorry. Ben was trying to ask a question.
Yeah, sorry.
And then Jeff, you'll repeat it.
I'll repeat it. Yep.
So I know we've only got a couple minutes left here. Just a question, I guess, with regards to allogeneic versus autologous therapies. Allogeneic has been a little bit shaky more recently. What would you say has changed with regards to your approach? What really makes it stand apart from maybe some of the challenges that Allogeneic has been having recently. And maybe I'll get you
so
to repeat that.
I'll repeat. Yeah, I'll repeat. So there's been a little bit of hesitancy for allogeneic T cells recently. I'm not keen to that, Ben. So I'll probably ask you after what you mean by that. But allogeneic T cells may be on, you know, a little bit of the hot seat. What do you think? I guess we can ask you two questions, or I guess maybe three. One, do you agree? Two, why do you think that is? And three, if you agree and that's the case, what is different about your technology that would make you avoid that?
Yeah. Go ahead, Munir.
To partially, we agree, because allogeneic T cells haven't fulfilled their purpose. They have done a great results, but the problem was the durability. So any effect, any response rate that you have seen in the patient didn't survive for long. And that's the limit. And that's why everyone, when the allogeneic hive of the T cells has started, everyone was jumping on it and expecting from it to be at least as good as the autologous CAR Ts. But they didn't deliver. And mainly not because they weren't able to kill. They have a great results in terms of killing, but they haven't been, the problem was the durability of the cells. And there was no problem in terms of processing, scalability, or production. The problem was the durability. And here where we differentiate ourselves in terms of solving the problem of the durability.
Yep.
We agree, yes, that the allogeneic T cells didn't deliver what it promised to do, but we all know from all of the clinical data, even the preclinical data, the problem is the durability, which we are solving. Of course, to additional modification that we have made to make sure that our cells will work in the hardest conditions, even the hardest and the harshest solid tumors.
Yeah. So I actually one of my like beefs that I had with allogeneic T cell therapies was you needed to make so many edits to these things. I mean, they needed to be just like edited up, you know, out the roof to have any kind of you know, activity. And that was one of the things. It kind of goes to my beef with in vivo CAR T too. It's kind of in vivo CAR T is you get one shot at this, like one edit, you're that's all you got. And it seems to me like one of the reasons that people were editing the heck out of these cells was because they were trying to figure out why are they not persistent. And the lowest hanging fruit is rejection. We just need to like edit the heck out of them and defend against macrophages and NK cells and T cells and all these and antibodies and just have 8 different edits, you know, to try and get it. And it's like we throw the kitchen sink at this thing and we can't get them to stay around.
No.
So yeah, that's I'm very interested to see if you guys have kind of cracked the puzzle here, because
and it's actually interesting that you mentioned the craze around in vivo CAR T. Because if you make in vivo CAR T cells, you will the exhaustion phenomenon will not go away. So
don't get me started on in vivo CAR Ts. I will go off for half an hour on it. I'm not a believer yet. We'll just put it that way.
Yeah. It may have a place in autoimmune, where an immune reset has the potential to give a cure. But we are also very much not convinced about the application in cancer.
Yeah, I tend to get myself in trouble when we talk about in vivo CAR T, because I'll just really just bash it. So we'll move on.
Very good. Well, I mean, we're right here at the end of the hour. Jeff, I'll kind of get you to take over here just so Munir can hear as well. But yeah, I guess maybe just in terms of next steps, like where are you guys planning on going next? What are the next big milestones for using this platform?
Yeah. So next big steps. What's the future look like?
Yeah. Next big step is a GMP manufacturing process. And we have thoughts about optimizing the manufacturing so it becomes really cost efficient. And then going into first-in-man clinical trial. And we have ongoing discussions with major cancer centers, both in the US and Europe, about this. And we hope to be in the clinic in 18 months' time to treat our first patients.
Awesome. Well, Jeff, again, I'm going to have to get you to do the outro so Munir can hear, but thank you everyone for coming today.
It's my first one. I'll take over.
It
So
is.
thank you for coming today. We appreciate your questions. Thank you, Rainer and Munir, for showing up. We'll get you to hang around for a little bit after the stream ends to make sure your video uploads. But yeah, until then, over and out.
Thanks, everyone.
Thanks, guys.
Thank you very much.